Hello everyone. Today we'll be discussing amelogenesis. Amelogenesis is the process of formation of enamel. So let's get started. So the formation of enamel occurs during the advanced bell stage of the tooth development. Okay, after the first layer of dentin is being laid down. So this is the diagrammatic representation of the advanced bell stage tooth bird. See here, this is the first layer of dentin which is being laid down. Now after this pre-dentin layer is being laid down, what would happen? The enamel matrix secretion would take place. So this enamel deposition, it begins on the cusp tip. Okay, the entire process is under genetic control. It begins on the cusp tips or the incisal edges, and from here it progresses outwards and cervical. Okay, the rate at which enamel is deposited is 4 micrometers per day, and the enamel formation, the rate of enamel formation is more in permanent teeth as compared to the deciduous teeth. The process of enamel formation or amelogenesis basically takes place into 3 steps. First, the enamel matrix would be laid down here. After that, there would be the mineralization of the enamel matrix, and then there would be the maturation of the enamel matrix. So this is a secretory ameloblast cell. First of all, we'll be discussing the formation of the enamel matrix. So enamel matrix formation begins or is being done by the secretory ameloblast cells. These are the cells which are well suited for the synthesis and secretion of the enamel protein. So when does the enamel matrix deposition begins? So it happens during the formative stage of the secretory phase of life cycle of ameloblasts. See, we have already discussed the life cycle of ameloblast. So during the formative stage of the secretory phase of life cycle of ameloblast, the secretory ameloblasts, they help in the synthesis and secretion of the enamel matrix protein. So how does the enamel matrix protein secretion begins? The secretory ameloblast has attained a certain polarity of the cell. In the distal or the secretory end, Golgi complex is present, whereas opposite to that, the nucleus and mitochondria are present. Okay. What happens is the messenger RNA. This is the messenger RNA. This carries the message from the nucleus to the cytoplasm. And in the cytoplasm, what happens is the ribosome translates the message and the protein is being synthesized in the rough endoplasmic reticulum. Now, the protein undergoes post-translation modification in the Golgi complex. After that, there is packaging of protein into the secretory granules. Now, what happens is these secretory granules, they fuse with the plasma membrane of the secretory ameloblast, and then they are exhaled out after the layer of pre-dentin is formed. Okay, so this is the process of exocytosis, and then see the pre-dentin layer, this pre-dentin layer would be there, after which the secretory granules would be laid down. As the matrix deposition, so this is the pre-dentin layer, and here in this region the enamel matrix protein would be synthesized. So as the enamel matrix is being deposited, the ameloblasts move outward away from the matrix. Okay, see, like this. And to have a contact with the enamel matrix so that their mineralization and maturation can take place, they lay down Tom's process. See, this is how the Tom's process is being spread up by the or laid down by the secretory ameloblast cells so that they can contact each of the enamel matrix proteins. Now, in the early stages, see the enamel matrix, it does not contain any collagen or any type of keratin protein. Okay. The enamel matrix in the early stages of amelogenesis, there is only 20 to 30% of the proteins which is present in the enamel matrix. And there are two types of proteins present in the enamel matrix, namely amelogenin and the non-amelogenin protein. The protein synthesis or the amount of protein content in the enamel decreases as they progress towards maturation via mineralization. Now, we'll be discussing both the proteins in detail. First of all, the amelogenin protein. So, the amelogenin protein, see, this is the enamel matrix which is formed, and we'll be discussing amelogenin protein. This constitutes the 90% of the total protein content of the enamel matrix. Okay, this is a hydrophobic protein and it has high content of proline, whereas non-amelogenin has less amount of proline present. It is present usually in the intercrystalline spaces. Okay, see here, represented by blue color. See, this protein, it is the gene coding for the amelogenin protein is located on X and Y chromosomes. So it varies in males and females, and hence it helps in identifying the sex of a person. Now, what happens is during there is the extracellular degradation of the amelogenin by the matrix metalloproteinases. Okay, so what happens is it is converted into small molecular weight fragments, which are the tyrosine rich amelogenin protein and leucine rich amelogenin polypeptide. So these tyrosine rich amelogenin protein and the leucine rich amelogenin polypeptide, they help to regulate the crystal growth. In a fully formed enamel, after maturation and mineralization of enamel has been taken place, in a fully formed enamel, there is a thin layer of amelogenin protein present, which is surrounding the crystal and also in between the crystals. Okay. If there is absence of amelogenin protein or any faulty amelogenin protein is present, then the enamel would be hypoplastic enamel. Next, if we talk about the non-amelogenin protein, this constitutes or this makes up 10% of the enamel matrix protein. It is a hydrophilic protein. It has high amount of glycine and less amount of proline. See, it envelops individual crystals like this. Okay, it would be enveloping the individual crystal. Now, the various non-amelogenin proteins are sheathulin, enamelin, ameloblastin, tuftelin, enzyme proteases, and amylotin. We'll be discussing each of these in detail, what are their functions or what are their uses. So enamelin, it is made up of amelogenin breakdown products, proteolytic enzymes, and serum albumin. The enamelin and ameloblastin, they together help to regulate the nucleation and the growth of the hydroxyapatite crystals. Tuftelin, it is present at or it is secreted into the DEJ. It helps in cell signaling, that is induction and reciprocal induction, which we have discussed in the development of tooth. Then enzyme proteases, they help in degradation of the amelogenin. So they facilitate its reabsorption during the maturation. Next is the amylotin. It is the amylotin protein. It is present in the mature ameloblast, secreted by the mature ameloblasts, and it helps in enamel formation. If there is absence of non-amelogenin protein, then no structural layer of enamel is being formed. Okay, now next is the development of Tom's process. How is the Tom's process formed? See, what happens is, after a little thickness of the enamel matrix is being deposited, the ameloblast develop the blunt conical processes at the distal end, and it penetrates and enters predentine. Okay, these processes, they are separated from the body by incomplete septa or the terminal bars, which are formed by microfilaments and tonofilaments. The cytoplasm of Tom's process does not contain any organelles. Instead, it contains secretory granules, some microtubules and microfilaments, and few mitochondria. Now, with the formation of with the development of Tom's process, the dual egg area of secretion of the secretory ameloblast becomes operational. This we'll see in the enamel rod formation. Now, what happens is the basal portion of the ameloblast cytoplasm has numerous secretory granules which are packed with the enamel proteins. These secretory granules come to the Tom's process, cytoplasm of the Tom's process, and then they are deposited or secreted out by the process of exocytosis. The Tom's process have fine radiating actin filaments, okay, which are extending into the cytoplasm, forming a web-like structure. Next, if we talk about the function of the Tom's process, then it controls the substance which would pass between the ameloblast and the enamel. Okay, this would control the substance passing. Along with that, because after its formation, what happens is it lengthens and narrows. Okay, what happens? As soon as the enamel matrix would be deposited, the Tom's process, the ameloblast would move backwards, upwards, and this Tom's process would lengthen and would get narrowed down due to the crystal formation. So the angulation of the Tom's process is significant in orientation of the enamel rod. Tom's process angulation gives the direction of the enamel rod. Okay, enamel rod ka direction is directed or decided by the angulation of, it is very significant in deciding the angulation of the enamel rods. If the Tom's process would not be formed, then what would happen? The enamel rods would be disoriented and there could be easy degradation of the enamel. Now we'll be discussing the formation of the enamel rod, how the enamel rod is formed. See, enamel secretion basically takes place through two sites. Okay, let us see how. See, the ameloblasts, secretory ameloblast has proximal junctional complexes and distal terminal bars. Okay. Now, the enamel rod secretion, we know that it takes place through two sites. One is this, the site 1, and this one is the site 2, from where the inter-rod substances would be secreted. See, this is the inter-rod growth region and this is the proximal end of the Tom's process, which is the inter-rod growth region. So here the inter-rod substances would be secreted. This site is located adjacent to the distal terminal bar all around the cell, and it results in accumulation of the enamel matrix between the adjacent Tom's process. Okay, this results in the formation of the inter-rod enamel. Now, what happens is a pit is created by these inter-rod enamel, and in this pit, what happens? The rods are secreted, enamel rods. This is the rod growth region, which is from the distal process of the distal portion of the Tom's process. So here you can see that the rod is being secreted. Next, what is there? Prisma sheath. It is a sheath-like zone and bordered between the rod and the interrod enamel. Okay, and this region is slightly more concentrated in the organic matrix. Thus, Tom's process is responsible for the rod-like structure of the enamel rod. Okay, of the enamel. So what happens is, first the interrod is secreted, interrod area. After that, a pit is created. Then in that pit, the rod comes, and between them is the prisma sheath, which is slightly more concentrated in organic matrix. Okay, now each enamel rod is contributed by 4 ameloblasts. Let us see how. See, these are the 4 ameloblasts numbered 1, 2, 3, 4. Now, the ameloblast 1 numbered as A1 would contribute in the formation of the head of the enamel rod. Okay, and then 2, 3 and 4, that is other 3, would lead to the formation of tail. So this is how the enamel rod secretion takes place, and one enamel rod is contributed by 4 ameloblast cells. Now we have seen how the matrix formation takes place. After that, the mineralization of the laid matrix and the maturation would take place. Now between the mineralization and maturation, there is a transitory stage. Okay, let us now talk about the mineralization of the enamel matrix, how the mineralization takes place. Now the mineralization process of the enamel matrix, it requires alkaline medium, okay, and it takes place extracellularly. Okay, so what happens is, see, what actually happens is, during the enamel formation, the calcium requirement for the mineralization reaches, see, this is the growing tooth bird, this is the circulatory fluid. Okay, so the calcium, these are the calcium molecules. The calcium required for the mineralization process, it reaches to the tissue fluid. Circulatory fluid, tissue fluid. So calcium is transmitted into the tissue fluid. After that, it reaches into the cell, ameloblast cells, and into the ameloblast, it gets binded, or it gets bound to the calcium binding proteins. See, these are the calcium binding proteins to which the calcium attaches. Now this calcium binding protein complex, it moves towards the distal cytoplasm. Okay, and they release their free calcium ions into the enamel matrix. See, distal cytoplasm mein aaye, the calcium ions are released into the Toms process. Now from the Toms process, what happens is, there is the calcium ATPase present in the plasma membrane of the Toms process. Okay, these calcium ATPase, what do? They extrude the calcium into the enamel matrix. So this is how the mineralization process would take place. Now mineral deposition in the enamel matrix occurs in 4 phases. The phase 1 is the primary mineralization, then secondary, then tertiary and the quaternary stage, or the third and the fourth stage. Okay. So in primary mineralization, what happens is, the enamel matrix undergoes immediate partial mineralization. Now see, the mineralization of the enamel matrix, what happens is, at the DEJ, the calcium or the mineral component of the enamel matrix, they are laid down perpendicular. See, this is the predentine, this is the enamel matrix. Non-amelogenin proteins, what do they do? With the help of the non-amelogenin proteins, the mineral content of the enamel matrix would first of all grow perpendicular to the DEJ in a needle or ribbon like pattern. Okay, so there is a needle or ribbon of the minerals in the primary mineralization stage. Then secondary mineralization, it starts at the surface and it proceeds towards the dentino-enamel junction. After that, the third stage, there is rebounding of the minerals from the inner layer of the enamel outwards. Then at the fourth stage, there is increased deposition of mineral in the enamel surface, which is why the surface of the enamel is hypermineralized. Okay. So what is basically the transition stage? It is the changes occurring in the ameloblast after the secretory stage and prior to the onset of the maturation process. What are the changes occurring in the ameloblast now? So what happens is the ameloblast cells, they reduce in height. You can compare these, okay? Next, the enamel secretion is completely stopped. The process of amelogenin removal starts. Half of the ameloblast cells, they undergo apoptotic cell death, which is the programmed cell death. The organelles which are involved in protein synthesis, they undergo autophagocytosis. The ameloblasts, they deposit a layer of basal lamina to attach themselves to the enamel, and these ameloblasts, they themselves are attached to the basal lamina with the help of hemidesmosomes. Next, there occurs a modulation or morphologic alteration. Now, what is this modulation or morphologic alteration? See, what happens is, in apical cytoplasm of ameloblast, there is an alteration between the smooth bordered ameloblast and the ruffled border of the ameloblast. Okay, every 5 to 7 hours a day. This one is the smooth bordered ameloblast, and this is the ruffled border ameloblast. Now, what are the differences between the smooth ended and the ruffle ended ameloblast? So, the ruffle ended ameloblasts, they contain mitochondria and they have villous surfaces which are packed with the mitochondria. Okay, so the proximal junction in the smooth ended is tight, whereas in the ruffle ended it is leaky. The distal junction is leaky in the smooth ended and it is tight in the ruffle ended. There is less amount of endocytic activity, whereas the ruffle ended shows more amount of endocytic activity because they contain numerous lysosomes. Next, the smooth-ended, they leak some amount of protein and water, whereas the ruffle-ended, they introduce inorganic component, that is the calcium, into the enamel matrix. Okay. Now, one thing we need to appreciate is the ruffle-ended ameloblasts, they secrete bicarbonate into the enamel matrix to keep the mineralizing front alkaline, and this prevents the acidification, thereby there is continuation of the mineralization process. Now let us talk about the maturation. How do the ameloblasts mature? So what happens is the maturation process is characterized by gradual completion of the mineralization. Here, about 90% of the initially secreted matrix protein and water is reabsorbed by the ameloblasts. The last layer of enamel, as I told, it is not formed by the Toms process, so it is the aprismatic enamel. Now there is rapid crystal growth which takes place during the maturation process. See, earlier the crystals were ribbon shaped. The mineral component were laid down in ribbon or needle-like pattern of 1.5 micrometer. There is rapid increase in the thickness, then width. So a plate-shaped structure is formed, which is around 25 microns, and they have high mineral content, which is the mature, and thereby they are called the mature enamel, plate-shaped structures. Okay. The amelogenin proteins are removed from the mineralizing front. So this is how the amelogenesis takes place into 3 steps, which is enamel maturation, enamel matrix formation, then mineralization, then a transitory stage, and then the maturation process. Now do not forget to like, share and subscribe to my channel. Do hit the bell icon if you don't want to miss the latest updates.